In this video, we will discuss about role of E-cadherin in invasion of cancer cells. So basically, they will check the E-cadherin impact on the invadopedia of the um, cancer cells. These cancer cells are usually developed invadopedia that can degrade the ECM surrounding them. Sorry about it. And that helps the cancer cells towards invasions and migrations. However, the role of E-cadherin before that was not too much explored. So first, what they did is they checked the presence of E-cadherin in the invadopedia and also in the intercellular parts. So they use these BXPC3 cell lines, and when they check it in the intercellular part, they are able to see the E-cadherins are present, and also these actins are also present. And when they check their overlap, they are able to see that E-cadherin and actins are overlapping each other, indicating that they are co-localized at a positions. They are co-localized in the intercellular part of the BXPC3. When they check the E-cadherins levels in the extracellular parts, they are still able to see the same thing, and they find the similar results, that is actins and E-cadherins are overlapping each other, and this invadopodia usually forms the layer of actins. So this indicates that the E-cadherins are present in the invadopedia regions. However, when they check the p-cadherins levels, they do not find the presence of p-cadherins in the BXPC3 cell lines. Here we can see that they are not overlapping each other, and the signals for the p-cadherins are also minimum. After this immunostaining, they also validate the similar results in the cell body membrane and the invadopedia fractions using the Western blot, and they are able to see the similar results, that is E-cadherins are present in the invadopedia sections, whereas p-cadherins is absent. So they have used p-cadherins and also this histone H1 as a negative control. So after validating that in the BXPC3 cell lines, which is the hybrid cell lines that has these E-cadherins in the normal form, and they, after validating these in vitro cell lines, they also check the patient samples, that is PDA, and the patient names that they called it as PDA-CO1T, and here they also find the results that actins and E-cadherins are present in the co-localizations form and in the invadopedia regions, and we can see that their gray values that indicates their overlapping regions of the actins and E-cadherins. So this patient samples also validated their in vitro findings that E-cadherins are mainly present in the invadopedia and the p-cadherins are not present in the invadopedia regions. And when they use this some 149 cell lines to, and this is the breast cancer cell lines, so this, in this case also they find that actins and E-cadherins are overlapping each other. So from this they made a point that the invadopedia's E-cadherins roles in the invadopedia is not only limited to the pancreatic cancer cells, but these things are found in the other cancers, and they have shown the evidence in the metastatic cancer cell line, no, this breast cancer cell lines. And here is the gray values where we can see that E-cadherins and actin levels are overlapping to each other. So in this video, they only made a point that although these BXPC3 cell lines are capable of expressing both E-cadherin and p-cadherins, because it is the E-cadherins that is mainly found in the invadopedia regions, and the p-cadherins are absent in that regions. And in our further videos we will discuss how these E-cadherins helps the cancer cells towards the invasions, and how there is a switch occurs from this E-cadherin is the marker for the epithelial cells. So how there is a switch occurs from epithelial cells to mesenchymal cells, that we might also cover in our subsequent videos. So I hope you understood this results that are shown here, that made a point that E-cadherins are present in the Invadopedia and P-cadherins are not present in the Invadopedia. That's all for this video. If you need to understand more about these experiments in detail, you can go to the reference that has been provided in the description. Thank you for your time.